next we will see about how some of these biomolecules comes in amino acids. So there are basically 20 different amino acids are there. So 20 different amino acids are there, and so 20 different amino acids, all these amino acids can be classified in different ways. So some based on side chains, we will see how they are classified based on side chains. If the side chain is going to be aliphatic side chain, we will see glycine, alanine, valine, leucine, isoleucine, like these will come in increasing order. So every amino acid will have CH, NH3 plus COO minus, that is the zwitterion state, and if it has that side chain as hydrogen atom, that is a very simple molecule, that is glycine. Its molecular weight is 75 and isoelectric point is 5.97. Isoelectric point is 5.97. Isoelectric point we will cover in some other video, and the hydropathy index is 0.4. So these technical terms can be covered in another video, so just these are for information. These amino acids differ in some molecular properties. For that information only this is being shared. Alanine means 2 amino propanoic acid. So 2 amino propanoic acid means like CHNH3 NH3 plus COO minus is linked to CH3 methyl group. So in the second thing there is an amino group. So in first case in glycine there is a hydrogen group, in the second case there is a methyl group. So 2 amino propanoic acid. Valine means 2 amino 3 methyl butanoic acid. So there are like four carbon atoms are there. In the second place there is an amino group, and the third thing there is a methyl group. How 2 amino 3 methyl butanoic acid? This is based on IUPAC naming. IUPAC naming. So IUPAC naming, we get this name, 2 amino 3 methyl butanoic acid. And leucine means like 2 amino 4 methyl pentanoic acid. So instead of butanoic acid there is a pentanoic acid. There are 5 carbon atoms in the main chain, and the methyl group is found in the 4th position, and there is an amino group in the 2nd position. Isoleucine has 2 amino group in the 2nd position and methyl group in the 3rd position, and it is a pentanoic acid as well. Isoleucine and leucine are isomers. Then we have, based on side chain, again hydroxyl group. So 2 amino 3 hydroxyl propanoic acid means we have serine. 2 amino 3 hydroxy propanoic acid is serine. 2 amino 3 hydroxy butanoic acid is threonine. And side chain, if it has sulfur atom cysteine, 2 amino 3 mercapto propanoic acid, so SH. We see that hydroxyl group is OH, and then sulfidyl group is coming here. Sulfidyl group is present in the 3rd position. So if here we are calling the sulfidyl group as mercapto, it's not as a sulfidyl we are calling it as mercapto. So 2 amino 3 mercapto propanoic acid. And then we go to methionine. Methionine is 2 amino 4 methyl thio butanoic acid. So since here in the AS sulfur group there is a methyl group which is attached, so we call as 4 methyl thio. So methyl is attached to here. So in the 4th position, methyl thio group, it is called as a called together methyl thio group, it is called as. So 4 methyl thio and then butanoic acid. And when we want to calculate the atomic weight, when we want to calculate the molecular weight, we have to remember the atomic weights of each individual atoms. Carbon has atomic weight of 12, hydrogen has atomic weight of 1, nitrogen has atomic weight of 14, and oxygen has 16. For example, in the case of glycine, if you want to calculate the molecular weight means we will say 12 into 2 plus 2 into 4 plus 16 into 2 plus 2 into 1. So we will get around 75. So this is how the molecular weight is calculated. And aspartate, next 10th one is, if you see, aspartate is 2 amino succinic acid. So succinic acid can be told as propane acid. So 2 amino propane acid. So when we write propane acid, we will know that CH2 CHNH3 plus COO minus, and then this side also COO minus is there, carboxylic group. So here I have illustrated the how the molecular weight is calculated. And then glutamic acid. Glutamic acid can be told as 2 amino glutaric acid. So 2 amino glutaric acid is nothing but 2 amino pentane dioic acid. So molecular weight is calculated. The unit of expression for the molecular weight is Dalton. So molecular weight of glutamic acid is 147 Dalton. Next we will go to, we have seen side chains which has alkyl groups. Next we have seen side chains which has hydroxyl groups. Next we have seen side chains which has sulfur atom. And then now we are going to see, then we have seen side chains which has acidic group. Aspartate and glutamate are acidic group. Next we are going to see side chains which has basic groups. So basic groups means which has the pH level on the opposite side of acidic one. So argin, the first case is arginine. Here the 2 amino 5 guanido valeric acid. So valeric acid is nothing but pentanoic acid. So uh, we have pentanoic acid. So we have 5 carbon atoms in the main chain. We will arrange the 5 carbon atoms in the main chain, and the amino group as usual it comes in the second position, 2 amino. And then guanido group, the fifth position there is a guanido group. This guanido group only we saw previously in the functional groups, we saw about that in the previous one. So guanido group is NHC, double bond NH and NH2. So the carbon is surrounded by 3 nitrogen atoms. So that guanido group comes in arginine. And next we will see about lysine. So lysine is 2,6 diamino hexanoic acid. So 6 carbon atoms are there, and the second position there is an amine group, and then sixth position there is an amine group. So there is 2 amino groups. So this is a basic one, NH3 plus. It has acquired hydrogen atom, extra hydrogen atom, thereby it has got an basic property. And then hydroxylysine is an uncommon one. So if we like to know its IUPAC name, it is 2.6 diamino 5 hydroxy hexanoic acid, and it is found in collagen. Histidine is 2 amino 3 1H imidazole 4YL propanoic acid. So this is structure. Propanoic acid, 3 carbon atoms are there, and the second position there is an amine group, and the third position an imidazole group is there with fourth imidazole group is there, and in the fourth position there is an YL, and I'm not sure about that why that YL is there exactly. I'll just try to get it clarified and then put it in the description. And next we will see the aromatic ring, the side chains which has aromatic ring. So next phenylalanine has 2 amino 3 phenyl propanoic acid. Second position there is an amine group, and in the third position there is a phenyl group. So even though that is a benzene ring, it is known as a phenyl group. It is called as a phenyl group when it is getting attached to something, when it is as a side chain. So 2 amino 3 phenyl propanoic acid. Tyrosine 2 amino 3 4 hydroxyphenyl. In the fourth position there is a hydroxy group in the phenyl group, therefore 4 hydroxyphenyl propanoic acid, that is tyrosine. And tryptophan, TRPW, so 2 amino 3 in the third position there is a 3 indolyl propanoic acid. So indolyl group is has benzene ring along with pentane, cyclic pentane group. So this, there are around 11 atoms are there. So 11 atoms are there in the and in the one position there is a nitrogen. So if this is the case, this is an indol ring. This is an indol ring. So 2 amino, 3 in the third position there is a 3 indolyl proponoic 3 indolyl proponoic acid, that is tryptophan. And amino acids. So amino acids are proline and hydroxyproline or amino acids. So proline is known as 2-pyrrolidine carboxylic acid. Its structure is here like this. Uh, so there it has a nitrogen group in one position, and carboxylic it is present in the nitrogen, and NH3 plus and COO minus are present in a cyclic way, proline. So it's carboxylic acid is outside, whereas the nitrogen NH3 plus is within this cyclic ring. Hydroxyproline has an hydroxyl group attached to the proline bond, so that is hydroxyproline. Hydroxyproline is found in collagen, and this is hydroxyproline is known as 4-hydroxy-2-pyrrolidine carboxylic acid. And there are other amino acids as well. Those are gamma carboxyglutamate, which is found in prothrombin, and desmosine, which is formed from 4 lysine residues. And it is a big structure, it's a cyclic ring. It will be something like a hemoglobin structure, and it is difficult to draw, but we can get the link where we can get the structure in the internet. And if we consider the polar uncharged thing means, we will say as aspargine and glutamine, those have been left out previously, unfortunately, so I have drawn those things. Those are polar uncharged side chains. So aspargine and glutamine. Aspargine has a similar thing to aspartate, which we saw previously, and glutamine has also a similar thing to glutamate, except there is a amino group in both cases. Instead of carboxylic group, there is an amino group. So their IUPAC names comes correspondingly. And if you see how the pyrrole ring is, pyrrolidine is there. I had drawn the pyrrole ring. How the pyrrole ring is? Pyrrole ring is something like a diamond structure, and it is it has a diamond structure. It is not and it's not a complete diamond structure. It is just closed in the top, and it has a nitrogen at the bottom. So this is how the pyrrole ring is there. In this pyrrole ring, we are seeing pyrrolidine. From this one, the pyrrolidine names comes. So this is for reference, and thanks a lot for watching this video.